So anyways, when we talk about the risk factors associated with foreign pathogen spillovers, we talk a lot about the phylogenetic distance between ancestral and novel hosts and the geographic exposure risks associated with this. But both of these factors can be either exacerbated or mitigated by standing genetic variation for susceptibility to foreign pathogens within a population. And so one useful thing to talk about with this is resistance transitivity. So that is the correlation between a host's susceptibility to an endemic and a foreign pathogen. And if within a population, this trend is generally positive, then selection for increased resistance against an endemic pathogen will also lead to resistance against foreign pathogens. So this can help buffer a population against foreign pathogen spillovers. However, if there are genotypes within this range where you have a higher susceptibility to foreign pathogens than you do endemic pathogens, then this can create an ecological niche for foreign pathogens to spill over and persist within a population. So this is something that some of our collaborators, Michael Hood at Amherst College and his student Noah Lerner, have looked at in the flowering plant Silene vulgaris. And so they collected a bunch of plants from the field, and then they measured their susceptibility to both their endemic anther smut pathogen and a foreign pathogen that was closely related but shared no coevolutionary history. And so overall, you can see that they found this general trend of positive transitivity where susceptibility to both pathogens was positively correlated. However, there were a few notable outliers that you can see in the black dots, and these individuals were much more susceptible to the foreign pathogen than they were the endemic pathogen. So what this suggests is that there might be two separate mechanisms of resistance going on here, a general mechanism and a specific mechanism, where general resistance provides broad scale resistance against many pathogen types, and specific resistance provides very strong resistance against a much more limited range of pathogens. So our aim was to see if we could replicate some of these results with a theoretical model. So to do this, we made a two-locus model with both general and specific resistance. So for general resistance, we assume that it provides an intermediate level of resistance against a broad range of pathogens. So for instance, in plants, this could be a mechanism such as thickening cell walls. And also we include specific resistance, which is effective against only a single type of pathogen. And so with this, we brought this into a deterministic SI model based on the dynamics of anther smut. So our model is haploid with random mating and some level of recombination between general and specific resistance. And we also assume that we have a sterilizing pathogen that does not induce additional mortality and frequency-dependent transmission. So we ran this model, varying the costs in terms of fecundity, both for general and specific resistance. And when we run this with just a single genotype of endemic pathogen, we overall find a pattern of exclusion between general and specific resistance. So on these plots here, the x-axis is the cost in terms of fecundity of general resistance, and the y-axis is the cost in terms of fecundity of specific resistance. The colors that are more yellow indicate fixation, and the bluer colors indicate loss. So for general resistance, we see that we get fixation when the costs of general resistance are low, and the costs of specific resistance are high. Likewise, for specific resistance, when its costs are low, we see intermediate allele frequencies of specific resistance. But notably, there is almost no overlap between the two regions where we're maintaining these forms of resistance. Practically no parameters allowed for the coexistence of general and specific resistance. So this means that we could potentially be exacerbating the risks of foreign pathogen spillover when specific resistance can invade. And this is especially notable because one thing we know about specific resistance from empirical studies is that it can often be very difficult to detect costs of specific resistance. Sometimes it might not have a high cost. So the fact that when specific resistance costs are low, we lose general resistance could mean that might be hard to maintain general resistance in this framework in a real natural setting. But another thing we know about specific resistance is that it can impose strong selection on pathogens to evolve some form of evasion for specific resistance. And this has been formalized with the gene for gene model and has been shown in many empirical systems. So we're wondering, what if we bring in a second genotype of the endemic pathogen into our model? Will this allow us to retain a greater frequency of general resistance across a broader range of parameters? And finally, will this allow us to recreate some of the data that I showed earlier? So to do this, we introduced a second genotype of the endemic pathogen into our models. And likewise, like as before, we assume that general resistance is broadly effective. So this new genotype of endemic pathogen cannot evade general resistance, but it can evade specific resistance. And we assume that there is some cost associated with this. So it has a slightly reduced transmission rate relative to the original genotype. And when we do this, we find overall that you see a much broader range of parameters where we can maintain general resistance. So our simulations where we have both of the endemic pathogen genotypes are shown in these bottom two plots. And you can see that the range of parameters where general resistance is maintained is much broader. Most notably, it does not depend nearly as much on the costs of specific resistance. We're maintaining general resistance even when specific resistance basically has no costs. We also see intermediate allele frequencies across some parameters that was never observed before without this. So to push this a little bit forward, we wanted to see if we could recreate some of the data that I had showed earlier from Michael Hood's lab. And so in natural populations, it's very difficult to actually know the specific genotypes that you're sampling. Instead, you're just sampling the natural variation as it exists. And we can actually recreate this with our simulation. We can do this by running it to equilibrium. And of the equilibrium population, we can assume random mating and ask, what are the probabilities of each possible full SIB or half SIB family? And if we sample a particular genotype pair, what is going to be the average resistance both to the endemic and foreign pathogen for each of these full SIB and half SIB families? And if we do that, we can get the plot on the right, which is analogous to the plot all the way on the left. And overall, we recapitulate this general trend of positive transitivity. We also are able to maintain some of these outliers, as I described before, that are more susceptible to the foreign pathogen than the endemic pathogen. We can do this by maintaining individuals that have specific resistance, but not general resistance. And one really interesting thing that came out of this is so far, all of the results I've shown have been assuming some intermediate level of recombination between general and specific resistance. But it turns out that that is highly consequential to our simulation results. If we assume that the loci conferring general and specific resistance become tightly linked, then we actually see a reversal of this trend of positive transitivity, and we get negative transitivity. So what's happening here is that the genotype that only has specific resistance becomes a little more fit without recombination, because it's no longer having to pay the cost of loss of gametes to these less fit genotypes. Its fitness is boosted, so we're maintaining more individuals that are highly susceptible to the foreign pathogen and have a low susceptibility to the endemic pathogen. So overall, these results show that with a single endemic pathogen, general and specific resistance can be highly exclusionary, which can potentially increase a population's risk for foreign pathogen spillover. However, this trend is mitigated when we introduce coevolution, which allows for the maintenance of general resistance across a much broader range of parameters. And finally, if tight linkage evolves between general and specific resistance, then this could result in a potential evolutionary trap that will lead populations to be more susceptible to foreign pathogen spillovers. And with that, I'd just like to acknowledge a few people that have been tremendously helpful in this process. And thank you. I think I have a little bit of time for questions.
I have a quick question on consequences for agricultural systems over here on the left. So this notion of transporting plant seed across regions. So we typically think of spillover being the foreign pathogen coming in and the evolution of the pathogen occurring at a faster rate. than the host. And so specifically in agricultural settings, this notion of exchanging seeds between regions to buffer against local adaptations of pathogens. And I guess I'm curious of the metapopulation consequences in a sort of a diverse agricultural system where instead of the pathogen coming in, it's in fact diversity of plants coming is the thing that's moving intentionally due to humans from a management perspective. I think back to the rice talk several years ago.
Yeah, thank you. That's a good question. And that's certainly something that has driven a lot of this is agricultural systems. I'm less familiar with that literature, but I mean, there are two approaches that have been seen widely where you have the stacking of multiple forms of specific resistance, which is very common in agricultural systems. But lately, there's been a lot more emphasis on the so-called, I think they use the term durable resistance in the ag literature, where it's looking at these mechanisms that would be more akin to the general resistance I'm describing. And in that case, that should be a more resilient approach to spillovers if you're assuming mechanisms behind this that are not pathogen specific. And in plants, we have good reason to believe that some of these things, such as thickening cell walls, waxy cuticles, should be broadly effective. And in that case, I think this would apply well to the situations you described.
So we have a raised hand from the online audience. Is that do you want to message in the Q&A, or can I give you do you want to ask your question? You can unmute yourself and ask a question. Or it may want to be one of those accidental raised hands. Hands down. All right. Hands down now.
So in your model, you went from one pathogen to two, and there was a real change of behavior. What happens if you go to three?
That's a great question. So that's something we're actively working on right now. Specifically, I mean, there have been lots of models looking at this multi-locus gene for gene system where you have lots of different specific resistance types that can combine. And usually in that case, you see negative frequency dependence between the multiple specific resistances. So it's a little early to say now. We've been trying to incorporate general resistance into a model like that. And one thing you do, it seems to be pretty chaotic and dependent on the parameters involved. But one thing we're found is it actually can repress general resistance, although that's very early work. So I'm not super confident in saying that.